Okay. Hi, so we're going to talk about complement today. So complement is one of those things that I skipped over yesterday, part of the innate immune system. It is important clinically, and that's why I kind of wanted to talk about it in its like own separate PowerPoint. Okay, so the complement system, it is a part of the innate immune system, and if anyone ever asks you, you're going to say it's a part of the innate immune system. But you'll notice, and we'll talk about this in a little bit, it's also technically a part of the adaptive immune system. So remember, the difference is the adaptive immune system is like your B and T cells. It's not like automatically activated. It has to be induced by like one of those antigen presenting cells going to the secondary lymphoid tissue and then activating a B or T cell. But it's still technically a part of it, and we'll see why. But basically, the complement system is basically complement proteins, which are synthesized by the liver as zymogens. And zymogen is just like one of those words that you want to recognize. It's going to come up again in hemonc like next year. So a zymogen is any protein that it's inactive when it's produced. So when the liver's producing these proteins, they're being produced inactive, like they're inactive when they're produced, and they must be activated. And a lot of the time, zymogens are cleaved to like expose like an active part of it, and that's like how it becomes active. Okay, so a lot of the times these zymogens get cleaved, and that's what makes them active. So the clotting cascade, when you guys do that next year, that's also a series of zymogens. But zymogens are tricky, and we're going to talk about why they're tricky in terms of lab testing. And that's kind of something you can look for, especially with clotting next year. So 3 different ways that complement can be activated, as you can see here, 1, 2, 3. But the end product is always the same. You always get a formation of what's known as the MAC complex, which we'll talk about, but you do need to know these 3 like the 3 ways of activating it, and then kind of like the downstream effects. So these 1, 2, and 3, these 3 pathways are the classical pathway, the alternative pathway, and the lectin pathway. And again, the only difference is in terms of how they're activated, but at the end of the day, you're getting formation of the MAC complex. One second. Okay. So the classical pathway is activated by antigen antibody complexes, and this is actually technically why it's considered a part of the adaptive immune system, because antibodies are only really produced by B cells, which are part of the part of the adaptive immune system, which is right here. And so, oh, hold on. Can you guys make sure you're muted? Sorry, just for the recording. I don't want it to be all weird. Just triple check. Okay. All right, so antigen antibody complexes are what activate the classical pathway, and all you really need is 1 molecule of IgM or 2 or more molecules of IgG. And again, we're going to talk about antibodies tomorrow, but a good way to kind of remember this is GM produces classic CARs. So GM for IgG and IgM are the 2 antibodies that can initiate the classical pathway of the complement system. So this is kind of like how it would look. IgG has like, it's just like a normal Y-shaped antibody. So you have 2 or more of them, and that can stimulate complement. IgM is a pentamer, so it looks weird. It doesn't look like a normal IgG. It doesn't look like a normal antibody, you would think. It looks like literally like a series of like 5 like spokes, and that's what IgM is. So you only need 1 of those to activate complement. Okay, and again, this is why it's considered a part of the adaptive immune system. You need antibodies to stimulate the classical pathway. So, and again, antibodies only in the adaptive immune system. The alternative pathway is activated by PAMs. So again, I put like 3 common PAMs: LPS, peptidoglycan, flagella. Remember, like any of like viral or bacterial DNA, RNA can also be a PAM. Anything that other organisms have that we don't have is considered a PAM. And remember, PAMs are recognized by your pattern recognition receptors, your PRRs. The alternative pathway activation is kind of weird. It's basically, it starts with what's known as spontaneous C3 tickover. And I'll kind of go into what that means. It's kind of a weird thing, but basically you have like this complement protein C3, and it's just randomly always being broken down into C3A and C3B. And I'm going to talk about this on a future slide, but I just kind of want you to be aware of this. So this is just always happening normally, which is kind of weird because remember I said like, oh, there's zymogens, they kind of have to be like cleaved to be activated. For whatever reason, C3 doesn't follow that rule. And it's very low levels. It really is like low levels that this happens, but C3 can spontaneously cleave itself into C3A and C3B. You don't need to know the specifics of that, just kind of understanding that that's something that happens. And again, super low levels. It's not like an issue at all. It's like really low. But if now there's a presence of a PAMP, so now it's like C3 is spontaneously like ticking over, right, being cleaved. And now there's like a bacteria here with LPS that's going to initiate the alternative pathway. So it's kind of like a two-step initiator, but it's like that's why the alternative pathway is basically activated by PAMPs because of the C3 takeover, and it can actually bind to those PAMPs. That's gonna be what's like activates that pathway. And I'll show you. I have like a visual to help you see that. But so this is LPS. Here's like the best PAMP. The best example of a PAMP is LPS. I realized we didn't really talk about it yesterday, but LPS is basically on the surface of your gram-negative bacteria. Gram-positive bacteria do not have LPS, and it's basically just like it's a sugar. It's a sugar, but it's comprised of 3 components. There's a lipid A. This is like what's embedded into like the outer membrane, which is just basically like a second plasma membrane. Okay. Then there's a core portion, and then there's the somatic O antigen or the O antigen. That's kind of what it's recognizing. Okay. But the whole thing, that's what LPS is. Again, only gram-negative bacteria. But gram-positive bacteria have tons and tons of what's known as peptidoglycan in their cell wall. This is a gram-negative bacteria, so it doesn't have that much peptidoglycan, but if you can imagine a gram-positive, there's no outer membrane here. There's tons of peptidoglycan. Like this entire thing is made up of like these peptidoglycan strains. But again, you guys will do micro in the summer, but I just wanted to give you a little bit of a background of what you're looking at. Okay. And then the lectin pathway, the last pathway, this is activated by mannose on the surface of microbes. So mannose is not technically a PAMP. A lot of proteins and whatnot in our own bodies are glycosylated with mannose, but we have this thing known as mannose binding lectin or MBL, and it can actually bind to mannose on the surface of bacteria. It's able to recognize like bacterial mannose somehow. And mannose binding lectin, we did briefly talk about this yesterday. It's one of those acute phase proteins that, IL-6 is going to trigger the liver to produce acute phase proteins. And then this is one of the ones that you can get is mannose binding lectin. And we'll go into more detail about this. Okay, so now we kind of made our chart look better. So instead of just having 1, 2, 3, now we have classical, alternative, and lectin. And then some things happen, and then we get the formation of MAC. So the classical pathway, as we talked about, antigen antibody complexes are going to activate this pathway, and IgM is the most potent, but you can also have like 2 or more molecules of IgG that can also activate it. And yes, no other antibodies can activate this pathway. It's either IgM or 2 plus molecules of IgG. CRP can also activate the classical pathway. CRP is also one of those acute phase reactants, but that can also happen with the classical pathway, but it's usually going to be your antigen antibody complexes. The alternative pathway is activated by PAMPs, and then the mannose, like the lectin pathway, is activated by mannose on the surface of the microbes. Okay, so now if we kind of like go from these long arrows to kind of making it a little bit more descriptive of what's going on, classical, alternative, and lectin are all going to have their unique like little pathways. And then eventually you're going to get to a commonly activated complex. So you can see all 3 pathways are diverging onto like a common pathway, basically. And then it's following this common pathway to get to MAC. So what we're going to focus on now is like what makes each one of these unique. All right, so here's the classical pathway activation. So remember we talked about 2 plus molecules of IgG or 1 molecule of IgM activates it. So those are your antigen antibody complexes. Or as I just kind of mentioned, yes, CRP can also bind to the microbe surface, and then that can stimulate it. What this does, like how this activates the complement pathway, the classical pathway, is there's something known as C1, which is a complex of a bunch of different C1 proteins. So there's C1q, C1r, and C1s. And it basically looks like a bouquet of daffodils. Like that's how you might hear it described, like a bouquet of daffodils. And when you have antigen antibody complexes, it's gonna trigger C1 to come to the area. And this is something that's going to happen throughout, like when you guys are going through like second year of med school, like I really want you to think when you have a pathology state where there's going to be antigen antibody complexes, there's going to be complement activation. And this is why it always triggers C1 to come into like to kind of come and stimulate this process. Okay, so we're going to talk about hypersensitivities next week. And two of the hypersensitivity, like one of the hypersensitivities is triggered by antigen antibody complexes. That's what makes it this specific type of hypersensitivity. So you can imagine, even if I don't say it, there's always going to be complement in that one type of hypersensitivity due to those antigen antibody complexes. So it's like just going to be a thing that you're going to see like throughout all of like the rest of med school and life, I guess. So the lectin pathway, like I mentioned, a microbial surface is going to have these mannoses on it that can be recognized by mannose binding lectin or MBL, which is also one of those acute phase reactants. So it's like, and mannose binding lectin has MASPs on its surface, which stand for mannose associated serine proteases. I just like telling you guys what all these names are. So you're like, what are these things? But this is like the functional part of MBL. MBL, again, it also looks like a bouquet of daffodils. So you'll see that the lectin pathway and the classical pathway are very similar. They're very, very similar. So I kind of talk about them together. And they're even similar to the point where MBL looks exactly the same as C1. It really does look exactly like how C1 looks. So since we're going to talk about these together, we can kind of talk about the classical pathway. Like I said, IgM is the most important, like the biggest stimulator. IgM bound to an antigen is like the most potent stimulator of this pathway. So let's say this is IgM bound to some sort of antigen. That's going to trigger C1 to come. And then what this is going to do is when C1 is now like bound to this, it's going to cleave C2 into C2A and C2B. I'm going to say a lot of C words like right now, like a lot of see this, see that. So I apologize. I tried to make it as easy as possible to follow, but it's going to be a lot. But so this is enough to trigger the cleavage of C2 into C2A and C2B. And again, it's able to do this because it's kind of like the masks back here on MBL. These are like the active part of it. These are like the enzymes, right? They're serine proteases. This is like the active part of MBL. Similarly, these parts of C1 are able to cleave. They're like the active parts of C1. So that's what's kind of activating this cleavage of C2 into C2A and C2B. Over here with the lectin pathway, we have a bacteria with mannose on the surface, like a terminal mannose, which is going to be recognized by MBL. And then when MBL binds to it, the MASPs are going to basically cleave C4 into C4A and C4B. And something that makes this even more confusing is the classical pathway, like C1, can also cleave C4. It's great, right? But MBL can only cleave C4. So I tried to keep the color coding. So C1 can cleave C2 and C4 into C4A, C4B, C2A, C2B. Mannose binding lectin can only cleave C4 into C4A and C4B. That's just like a little difference between the two. Then what happens, once we have these nice cleavage products, we have C2B and C4B, they're going to actually come together and make a complex called C2B-C4B. I swear to God, this is how they came up with these names. So C2B, C4B, people call this C3 convertase. And I'm going to warn you, this is like commonly asked. It's like what is the makeup of the classical or lectin C3 convertase versus the alternative C3 convertase. So it's just, that's why I'm kind of emphasizing it. You should know like both of these names. You might also see anytime we talk about a convertase with the complement system, like C3 convertase, they could write it as like, so C2B, C4B. And they want you to know that this is a convertase. So what they do, and you might see this in like old textbooks, like they draw a line over it. And when you see this, so like C2B, C4B with a line drawn over it, this is a convertase. Specifically, this is C3 convertase, but that's just what that line over it means. It means it's a convertase. Okay, so we're here, we're following. Okay, so we need the B's. We'll talk about I think I put that. Yeah, we'll talk about what the A's do later. They do have roles, but right now the B's, the C2B and C4B come together and they form C2B, C4B or C3 convertase. All right, so let's talk about the alternative pathway. So the alternative pathway activation, remember, it's going to trigger, remember, we always have these low levels of C3 being converted to C3b. That's what's known as spontaneous C3 tickover, as I mentioned. And in the presence of something like a PAMP, like LPS, which is just that sugar moiety, you can get alternative pathway activation. So the alternative pathway, for example, here's LPS, here's your spontaneous C3 tickover, and now we're gonna activate that pathway because it's doing it in the presence of a PAMP. So once we have C3b, that's part 1 of the alternative pathway activation. Part 2 of it is we have more factors. So we have what's known as factor B and factor D. Factor D cleaves factor B into factor BB. Swear to God. At least they're trying to keep it consistent, right, with the B's. But okay, so these are the 2 things. So we have C3 spontaneously converting to C3b, and in the presence of a PAMP, this is going to trigger this part of the pathway. Then factor D is going to cleave factor B into factor BB. And you know what's coming. We got a C3 convertase. But now the C3 convertase for the alternative pathway is C3b BB. I know. Shake my head. So you can also see this written as like I said with the last one, C3b BB with a line over it. This is your C3 convertase for the alternative pathway versus C2b C4b for the classical or lectin pathway. And the order of this doesn't matter. Like if you see what did I write first? If you see C4b 2b, that's fine. The order does not matter here. Same here. If you see BB, C, they probably won't do that, but you can rewrite this and have the BB, this part first. It doesn't matter. Okay, so that's how we get to the C3 convertases. So now let's go back to the classical and the lectin pathway. So we're here at this point where we have our C3 convertase made, and now, what do you know, we're gonna get conversion of C3. So this is kind of what happens in both of these pathways. So now these pathways kind of already divulged, which is good. You don't have to memorize too much different between these 2. So at this point, we have C3 now is going to get converted into C3a and C3b by C3 convertase that we just made. And then from here, which I think is really strange, but it's kind of cool that it happens, you're going to have, so C3 convertase is kind of like an enzyme. It's not like it's getting used up or it's not getting like used in this reaction. It's just kind of like being an enzyme, right? So it's actually going to combine with C3b. So now it's almost like a fail-safe mechanism. They're like, okay, we're going to move on to the next step if we have enough C3b. So C3b and C3 convertase are actually going to combine. It's kind of like, not really combine because this is an enzyme. C3b kind of just like sticks around the surface of it. But when that happens, now you have C5 convertase. And another way to write that is the same thing as before, C2b 4b, which is the C3 convertase part, now with this additional 3b on it. And that's your C5 convertase. So again, you could see C5 convertase, you could see C2b 4b 3b. They're the same thing. And you should know that makeup because it's going to be different from, obviously, from the alternative pathway. And that's why we're taking the time to like painstakingly go through this, because they will ask the makeups of these convertases. Okay, so that's C3 convertase, what it does, and then how you get C5 convertase from here. Going back to the alternative pathway, so we have our C3b C3 convertase. It's also going to convert C3, which is nice. So it converts C3 into C3a, C3b. And again, it's the same thing actually. So C3b, once it's made in a high enough number, it's going to combine with C3b BB with the C3 convertase, and we're going to get C5 convertase. So this is a really fun mouthful to say, but C5 convertase on the alternative pathway is C3b BB 3b. It's ridiculous. But yeah, so that's kind of like, again, the main differences is between these pathways is going to be like the exact makeup of the convertases, but they're all kind of doing the same thing. Okay, so from here, we just kind of talked about how the C5 convertase of the classical pathway and the lectin pathway is this specific C5 convertase. It's C2b 4b 3b, whereas the alternative pathway C5 convertase is C3b BB 3b. Okay, so from here, what they do, and I put both here, but they're doing the same thing. It's not like both of these are kind of happen. Like it's like it's based on what pathway you kind of activated is depending on what convertase you're getting. So C5 convertase converts C5, which is nice. So it's going to cleave C5 into C5a and C5b. And then C5b is not joining with C5 convertase, which is nice. C5b is going to be actually what kind of triggers the formation of the membrane attack complex, that MAC complex. So C5b is actually the exact thing that happens is C5b binds to C6, and then that facilitates like C7 coming in, C8 coming in, and C9 coming in. But it doesn't matter. It's just C5b is going to combine with all of these other C like complement proteins. So C6, C7, C8, C9. And this is what makes up the MAC. You should know what makes up MAC. And it's going to be the same in both pathways, or all three pathways. So MAC is made up of C5b through, a lot of the times they write like you'll see C5, B through C9. Or some people just do C5 through C9. That's fine. It's just they're not being as specific, but it's not a big deal. C5 through C9. You do need C5b in there, so like that would be more correct than saying like C6 through C9, because C5b is kind of what's triggering the formation of the MAC complex. Okay. All right, so what does MAC do? It attacks. That's why I call it MAC attack. So it attacks cells. So here's like the microbe. And again, this is non-specific. It's in the innate immune system. So it doesn't really care what kind of microbes it comes against. It's going to just attack anything that it can. And what it's going to do is once it comes in it, it's going to poke holes in the surface. It's literally like, and it just forms these pores. Like if this is not actually what MAC looks like, it looks like a giant pore forming like unit thing. And it inserts into the membranes of these microbes and it just pokes holes in it. And then like just water rushes in, they lyse. So that's what MAC doing. It's really smart. It's really just poking. It's not really doing anything. And that's why it's good, because it's a non-specific. This is a part of the innate immune system. It's non-specific. So you're able to actually just pretty non-specifically kill things that are present. So functions of complement, other to produce MAC. So MAC production is like the main function of complement. That's what we're trying to do. But what's really important, and this is what I mentioned we were going to talk about today, is the production of opsonins. So opsonins basically make microbes look more appetizing to phagocytes. So remember, some phagocytes are like macrophages, dendritic cells, neutrophils. Neutrophils are the best phagocytes. So like PMNs, macrophages, NDCs. Like these guys are going to go, basically they're always kind of going around. They're part of the innate immune system. So they're just kind of going around looking to see if there's anything wrong that they can eat up, basically. And what C3b does is it's just literally sticks on the surface of microbes, and it, for whatever reason, that makes these phagocytes be drawn to that microbe. It just makes them look really appetizing. That really is what they do. And this is really important, and I have a lot of things highlighted in yellow because it's all really important. And you're going to see when we're going through clinical relevance why this is so important. C3b, so opsonization in general, like C3b is the most important opsonin, but opsonization in general is so important to get rid of any bacteria that has a capsule. You guys remember those, right? Remember the three main ones I told you to remember? So we have our, strep pneumo. So strep pneumo is one. Then we have Haemophilus influenza, and then Neisseria meningitidis. And you'll see, we'll talk about this later, but like whenever I write Haemophilus influenza, type B, so that's why I have the B here, type B is the one that's encapsulated. There's a bunch of different types of Haemophilus influenza, but type B is the one that's clinically, like if you have a Haemophilus infection in like a kid in the future, it's usually going to be type B. That's what like the vaccine against it is for. It's for type B. But yeah, so you really need opsonization to clear out these encapsulated infections. So strep pneumo, Haemophilus influenza, and Neisseria meningitidis. And the most important opsonin is C3b. So that's basically like the second function of complement, and that's why it's really important. Those B's are the ones that are really, really important at opsonizing, especially C3b. You also get production of what's known as anaphylotoxins, and I don't mind this really obnoxiously long word because it starts with A, and this is all of those A molecules. So all of those A molecules are, all of them, they're anaphylotoxins, and all anaphylotoxins are are basically pro-inflammatory, like they initiate an inflammatory response. So some things we talked about yesterday, that's a part of the inflammatory response, mast cell degranulation. Remember, mast cells release histamine, which causes vasodilation, and it causes like the endothelial cells to get more leaky so things can go through. Then the neutrophil is going to come in from the site, and it's going to come through, it's going to go through margination, rolling, diapedesis, that whole thing. That's what we're kind of stimulating with the anaphylotoxins. So not only are we getting MAC, which is going to directly affect the organism, we're also getting all these opsonins being produced, which can also affect the organism. And we're getting anaphylotoxins produced, which can also affect the organism. A little more indirectly, but it's bringing in all the players you need. So the A ones are C3A, C4A, C5A. They're all anaphylotoxins. And then C5B, I think I highlighted this because this was on the study guide last year from Dr. Demarest, so I don't know how important this is to know, but it might come up. But C5b is also a neutrophil chemokine. So remember, chemokines are like basically cytokines that tell cells where to go. So if you have C5b, a lot of C5b being released, you're going to have a lot of neutrophils coming into the area, which, I mean, you're already doing with the anaphylotoxins, but like even more so now, because that's like what C5b's role is to call in neutrophils. So complement, we got MAC, we got opsonization, we got anaphylotoxins. And that's what makes complement such a important thing. Okay, so this was kind of like another thing that I wanted to mention. So we're getting MAC, which is C5b through C9. That's going to lyse the bacterial cells. We're getting C3b, which is like the best opsonin you can imagine. There are some antibodies that act like opsonins, which we'll talk about tomorrow. We'll talk about antibodies tomorrow. So some antibodies can also act as opsonins, but C3b is like by far the best opsonin that the body makes. C3b also binds to antigen antibody complexes. So you can imagine that we're already getting C1 binding to antigen antibody complexes, and then we're getting C3b binding to antigen antibody complexes. And why this makes sense is because we want these to be removed. And I know I used a red blood cell here as an example, but imagine that this could even just be like a C3, like it doesn't have to be a red blood cell. Like just imagine this is like a C3b receptor on like a macrophage or something. Like pretend this is like a macrophage. So macrophages have C3b receptors on them. And basically, they can bind to C3b on these antigen antibody complexes, and it can actually facilitate their removal even quicker. Because again, in a specific type of hypersensitivity, the antigen antibody complexes are what's causing a pathology state. So we want these out of the body. So as soon as we get antigen antibody complexes, we want them out. And that's kind of like another way we can do this. Yes, we're getting complement triggering, but at the end of the day, we're getting C3b to also bind to those complexes to kind of facilitate their removal. So red cells and B cells specifically have it's called CR1. I think, again, I highlighted this because Dr. Demarest wanted me to highlight. I don't think you need to know this receptor name. But I think it's good to know that red blood cells and B cells have a C3b receptor, as well as a lot of those phagocytic cells, like macrophages especially, because then they can go to the spleen, right? And then they can kind of like get picked off. Like you can kind of they can get picked off, and then you can remove them. Okay, so yes, C3b is an opsonin, but it's also going to bind to those antigen antibody complexes to help facilitate their removal. Then we get the production of those anaphylotoxins, so C3A, C4A, C5A, and we're going to get the production of C5b, which is a neutrophil chemokine. So I like this picture. It kind of shows, well, it's just showing C5b C5 specifically, but C5a, here's your anaphylotoxin. It's everything we talked about. It's just you're inducing that inflammatory response. And then C5b, it's a part of MAC, and it's like a producer. It like calls in neutrophils. It calls in neutrophils to the area. Okay, so notes on complement. So this is what I kind of wanted to talk about with zymogen pathways. This is going to come up again next year with coagulation. Zymogen pathways are very tricky, and you really need to take your time and read the question, because I don't want you guys to get got when you're reading these questions, because you feel comfortable with it, and then you just bypass this concept. If you have, I'm strictly talking really about like lab tests. if you notice that there's less of a zymogen, so less like lower numbers of like C4, for example, that means the pathway is going through very quickly. It's active. And the reason why is because you're using up the zymogen, you're converting it faster. Like lab tests are testing for this. They're not testing for C4A and C4B. They're testing for this. So if it's being converted into like C4B and C4A very quickly to the point where you have really low levels of C4, that means you're going through the complement pathway. So for example, we have tons and tons of microbial surfaces or tons and tons of antigen antibody complexes. You're going to consume complement. And when you're consuming complement, you're going to have really, really low levels. And they usually always look at C4. Technically, they can look at like, C2 or C3, but just for lab tests especially, they always tend to look at C4. I think it's because it can get activated by the lectin pathway or by the classical pathway. So this is like normal. We have tons of C4. Remember the liver just makes zymogens. Like they're inactive, but it makes them until like it's activated. So that's why we have normal levels of C4. But if we activate this pathway and go through the pathway, we're consuming C4, so we're gonna have very low levels of C4. I'm really emphasizing that because that's it's really easy to confuse that. And again, this is gonna come up with coagulation next year. It's the same exact concept. If you have less clotting factors, you're clotting. So how do you measure complement? It's known as a CH50 test, and it just basically it tests for C3 and C4. But I think that's just how the test was designed, that it mostly tests for C3 and C4. So again, if you have a low CH50 result, that means complement is being consumed. So disease, like this patient might have a disease state, especially those with antigen antibody complexes. And we're going to talk about lupus later, rheumatoid arthritis, any like really inflammatory like disease, like an autoimmune disease, anything that's tons of inflammation. Lupus and rheumatoid arthritis produce a lot of inflammation. You can imagine that the CH50 result is going to be very, very low because you get these antigen antibody complexes, we're going to get tons of complement activation. There's really like high CH50 is like not really a thing. It's a lot like less clinically relevant. It could mean the liver is like what I wrote, spazzing out and just making tons of acute phase reactants, but you really don't see a high CH50. It's either going to be like CH50 is normal or CH50 is low.
Christine, I have a question.
Yeah.
I don't know if I'm getting confused, but so when you say that there's a low CH50, you say complement is being consumed, does that mean it's active and like trying to fight this infection, or it's like it's being consumed by the bacteria? Do you get what I'm saying?
Yes. It's like consumed with the air quotes. It's like it's being converted. It's being converted to like C3, C4A and C4B. So we're consuming C4 when we do this. Yeah, it's like not really like the bacteria is not eating it. It's just like, that's like a term that I guess textbooks and whatnot, Dr. Ryan, I think will say this too. You're consuming complement, not the bacteria itself. It's just like you're converting it to something else. Yeah, it's a good question. Yeah, it's like not a literal consumption, but it's just like converting. That's like the terminology that they use. Okay. So now we have regulators of complement, and these are going to come up in some pathology states. So I kind of put them together. So there's some regulators that you should know. There's what's known as factor H and factor I, and you have to kind of keep these separated from factor D and factor B. I know they really could have came up with better names, but it's okay. Then there's what's known as a membrane cofactor protein, or MCP, and another name for it is CD46. And then properidin. So these are like the main regulators of this system, because you can imagine you don't want complement active if you don't need it to be active. So it is very highly regulated with a lot of the stuff in the immune system. So factor H, factor I, and MCP, this is what they do. So basically MCP is going to be, it's just kind of like, it's this yellow thing. This is MCP. So what can happen is C3b can also be bound by MCP to kind of regulate it. And then factor I can kind of bind to it. And factor I can kind of bind to it, and then it can basically make sure C3b remains inactive if it's not trying to be active. That's all it's really doing. And it can, factor I and MCP can similarly kind of do this with C4b. And again, just kind of make sure that they're remaining inactive if you're not trying to activate complement. But factor H, so here's kind of like this picture in like better words. So here's factor H. Factor H, it wasn't on that previous, it wasn't on here, but factor H is going to stabilize C3b. So again, remember C3 spontaneously ticks over. So we have a lot of C3b, well, not a lot. We have like low levels of C3b always being produced. But again, we don't want it to go accidentally activate the complement system if we don't need it to be. So factor H kind of comes in and it stabilizes it. Then MCP is going to bind to it. It was that yellow thing. I tried to like copy it from here. So MCP is going to come in and also kind of bind to it. MCP is a cofactor for factor I. And then here comes factor I. And then factor I is going to come in, and then we're going to get destruction of C3b. So back here, it's kind of like just factor H is not really on here, but that's what's happening. We're getting stabilization. We're getting MCP kind of like associating and binding to C3b, which is going to allow factor I to dock and then cleave it. So you don't have to know these cleavage products, but just know that it just basically keeps it inactive. So my thought question for you guys, and you don't have to answer this, it's just something to kind of think about. If you have a factor I deficiency, what do you think a CH50 test will show if you're looking at C3 levels? I'll give you guys a minute so you can like draw it out. It's kind of, these things are really confusing. Okay, so some of you guys are answering, which is good. So the term CH50, it's like an arbitrary number. It's not really arbitrary. It's supposed to represent complement levels at exactly like halfway used, basically. So it's kind of like you have a normal CH50 value. So if it's lower than that, that's kind of where that comes from. I forget what the H stands. It's like complement something at 50, at like 50% use. So it's kind of like, what is it with enzyme kinetics? Like the Km is at half Vmax or whatever. It's kind of the same idea. It's like we're at halfway between like what the total levels of C3 and C4 should be. And if we're less than that, then that means complement's being consumed, right? So I think that's like where the 50 is coming from. So why does factor H and I bind to C3b as opposed to C3 before it gets broken down? It's a good question. I think it's just because like it's just an evolutionary thing that adapted. Like you want C3 to spontaneously convert to C3a and C3b, right? Like that's how you initiate the alternative pathway. So you still want that to happen. So if you're having factor H and factor I bind to C3, maybe over time you wouldn't have gotten to like that. You wouldn't even have an alternative pathway maybe. So I think it's just kind of like a way to be like, okay, that can still happen, but we want to make sure that this remains kind of like not active, if that makes sense. That's what I think. I'm not 100% sure, but that's what I would think. They're trying to prevent like the more important one from doing anything. Oh, okay. H means hemolytic. Thank you. Yes, something, yeah. So that's, yeah. Thank you for looking that up. Okay, so let's go back to this. So if we have a factor I deficiency, what will our CH50 test show if we're looking at C3 levels? So factor I, the end of the day, it breaks down C3b, which is what we want. We want normal C3b levels. So if we have a factor I deficiency, now C3b can go do stuff. It can go do stuff if it wants. And if it goes to go do stuff, you're actually going to consume more of it. So the levels are gonna be low. You're gonna have a low CH50 if you have a factor I deficiency. Is anyone still confused by that? It's gonna be again, it's with Zymogens. Zymogens are really tricky to answer questions about, and you just kind of have to really think them out. So again, with Zymogens, this is gonna be, if you don't do anything with this, if you let it go, it's gonna go bind to like the C3 convertase, C2b, C4b, or what is it, C2b, BB, right? Like it's gonna go bind either to that pathway, like to one of the convertases, and be used up that way. So it's going to be consumed that way. And that's if you allow the complement system to kind of go through. You're allowing for C3b to be used up. So if you don't stabilize it and basically destroy it, it's going to be you're going to have really low levels of C3. And again, super counterintuitive, because you're like, well, you're destroying it with factor I. Shouldn't you have high levels? No. And that's why. It's because it's a Zymogen. Good. Yeah, I think a few of you guys did read it wrong. I just didn't want to sit here and be like, oh, factor I deficiency equals low CH50, because I think it's like kind of working through it is going to help. And you should just kind of like, when we're done today too, just kind of like work through like, well, I have some questions for you, but kind of work through these Zymogen-like questions. So if we get rid of basically a negative regulator of complement, we're going to allow complement to go. So we're going to have low levels of CH50, and CH50 tells you, if you have a low CH50 test, you're allowing complement to go, so to speak. Okay. So that's what these guys do. So all of these guys, I could have changed this to say factor H deficiency. I could have changed this to be an MCP deficiency. Any of these would kind of have the same result, because they're all these negative regulators of complement. Okay. So then what's properidin? Properidin is a positive regulator. So what properidin does is it's going to stabilize C3 convertase. That's really its only role. And honestly, if you saw an actual picture of properidin, it like sits on top of C3. It literally sits on top of it. And it's, again, just trying to make sure, like, we really want, like, since we got to this point, like, we made C3 convertase, we're going through complement. That's what's meant by like a positive regulator. Like, we're committed now to going through this pathway. So that's what properidin does. It kind of allows for the continuation of complement. So this doesn't just like randomly break down. So it like sits on it. I think of it as like it sits on it to kind of keep it like nice. So it doesn't just randomly fall apart. Like these multiple like C3b and BB don't just like fall apart. So that's what properidin does. It positively regulates the pathway. We want more properidin. Sorry, we want the complement pathway to proceed at this point. So, oh, good question. Oh my gosh, sorry I didn't mention that. Great question. So properidin only stabilizes the alternative pathway C3 convertase. Very good question. So properidin would only be found in to stabilize this C3 convertase. Whereas like factor H, I, MCP, whatever, they do any C3b. Okay, so then we have some inhibitors of complement. So this kind of goes back to the question yesterday about like red cells don't have MHC class 1, right? So like why don't they get attacked by NK cells? And it's because red cells have other things on their surface to kind of prevent them from getting like eaten by NK cells, or actually now here getting like attacked by complement. So there's a bunch of stuff that helps kind of inhibit the complement system. So two of them down here, it's called DAF and MERL. So DAF stands for decay accelerating factor, which is CD55. And then MERL stands for membrane inhibitor of reactive lysis, which I think that's a very good name for a complement inhibitor. So MERL, and that's CD59. You should know, and I know it's annoying, but you should know at least these CDs. I think the MCP CD is not as important, but these you should know. You will get a question on, like there's a pathology state where you're missing one of these CDs, and then that can lead to a pathology. So definitely know those, because it can come up in questions all the time. And what they do is kind of what's on here. So here's like a red cell, and red cells have DAF and MERL, neutrophils have DAF and MERL, and platelets have DAF and MERL. And it kind of, again, kind of prevents them from getting attacked from complement. DAF is going to prevent C3 convertase formation directly on the red cell. Like it's just going to stop it from like binding to it. And then MERL is going to block MAC. Like you're blocking the binding of C9. And remember, MAC, you need C5, C6, C7, C8, and C9. So MERL is going to block C9, so you won't get MAC. And then we also have what's known as a C1 inhibitor, which is, I think I just drew it as like a rectangle over C1. So here's C1 inhibitor. It just inhibits C1. And again, remember, C1 can activate complement by binding to antigen antibody complexes or even CRP bound to the surface of something. So again, it's kind of confusing because it's C1 inhibitor. So we're back to that concept of inhibiting complement. So I kind of wanted to draw it out like with the pathway. So C1 inhibitor is going to inhibit C1. So we're getting inhibition here. So we're not going to get this process to happen. Like we're not getting this process. So we're going to have normal levels of C2. Like we'll have normal levels of C2 with C1 inhibitor. But a side note of C1 inhibitor, and this is the clinically relevant part, C1 inhibitor facilitates the process of bradykinin getting degraded. So the degradation of bradykinin into just like byproducts. C1 inhibitor does this, which is really strange. And again, it's going to come up like it's a clinically relevant tie-in. So C1 inhibitor, the normal role is it inhibits C1. We know that, it's in the name. But it also facilitates the breakdown of bradykinin just in general, just breaks down bradykinin. I guess I should have put like an X over this or something. But it degrades bradykinin. It helps do that. So this is why it's important to understand what C1 inhibitor does for a pathology state known as hereditary angioedema. So hereditary angioedema, HAE, it's a hereditary deficiency of C1 inhibitor. Oh yeah, another name of C1 inhibitor is C1 esterase inhibitor. It's the same thing. So you can see C1 INH or C1 esterase inhibitor, they're the same thing. So we don't have that. So now like go in your head, be like, okay, so we don't have this inhibitor of complement. So start trying to kind of put these pieces together. Bradykinin, I don't know if you guys had talked about it in Palm, but one of the things that it can actually do is it can produce nitric oxide, and which you can imagine can cause like vasodilation and stuff. And it like causes some other stuff. But notice that ACE inhibitors, which I know you guys haven't had cardio farm yet, but ACE inhibitors inhibit ACE, and they also inhibit bradykinin breakdown. So they block the breakdown of bradykinin. So we won't have bradykinin breakdown. We'll have very high levels of bradykinin. I have no idea how common this is. On boards is pretty common. No, I honestly don't know. And we'll talk about like how you would kind of see it. Is it rare? To be honest, people don't know that they have this until they're on like an ACE like are you talking about hereditary angioedema, like the C1 inhibitor? Yeah. So they won't know that they have it until they're put on an ACE inhibitor, and then they have these symptoms. But I'm not entirely sure how common it is. But I know that that's like your classic pathology state. Like that's your classic vignette in the question. So it's not like it's gonna do anything too bad. You might notice something like not having C1. Like you might have complement being activated like a little bit more than it should. But like it's not that important. Like it's not that big of a deal if you don't have any. But what's gonna be the issue with this is when you put the patient on an ACE inhibitor. So you put the patient on an ACE inhibitor, now they're just going to have tons of bradykinin because first we don't have degradation of bradykinin, which is the normal physiologic role of C1 inhibitor. And now the ACE inhibitor is going to further block bradykinin breakdown and just complete. So we're going to have tons and tons of bradykinin buildup, and that's going to manifest as massive swelling. And then again, CH50 tests, we're having decreased C4 levels because C1 normally activates C4 being cleaved, right? So if we don't have this inhibitor, C1 is able to just go ahead and do that. So we're going to consume C4, and that's why you would have a decreased C4 levels if you do the CH50 test. So again, if I talk through that, because question. It's a good question. I don't know 100%. I feel like I used to. So the question is, why does bradykinin result in that type of swelling? I think I used to know, and I completely forget, to be honest. I'm not sure, but and you can look it up. They won't ask about it. They'll kind of like see if you understand what this pathology is. But yeah, I'm not 100% sure. I think it probably has something to do with it deposits and then draws in fluids, but who knows? But anyway, so so imagine there's something else that's working in this disease state that lessens bradykinin in the system. Otherwise, patients would have a swollen face even before the ACE inhibitors would give in. Yes, that's a good point. That is a good point. Or what could happen too is bradykinin could be shunted to the nitric oxide pathway, like in this pathology state before an ACE inhibitor. Like it could be shunted to another pathway. So there is probably something else that's going on to keep bradykinin relatively normal until you gave an ACE inhibitor. And then it's like, it can't, like the body's like, nah. I don't know what to do with all this bradykinin. But I kind of wanted, again, to talk about the CH50 test. Spoilers. But I wanted to talk about this result because, again, C1 inhibitor normally inhibits C1. So it kind of keeps everything normal until you need complement to be activated. If we don't have C1 inhibitor, so we don't have an inhibitor, we're going to get activation. It's like Dr. White says, if you inhibited an inhibitor, right? So we are inhibiting an inhibitor with the C1 esterase deficiency. So we're allowing for activation of this pathway. We're allowing for consumption of C2. We're allowing for consumption of C4. Because remember, C1 also does that long arrow over to C4. It can also activate C4. And the CH50 tests for, C3 and C4. So we're also going to get low levels of C4 because, again, C1 activates that process. So here is your buzzword B for hereditary angioedema. They will never tell you patients on an ACE inhibitor, ever. If they do, you're super lucky because they shouldn't tell you this. But what they'll say is they'll give you like a whole vignette about someone having really bad hypertension. And then they'll say the doctor just switched him over to a new medication, which you're like, it's an ACE inhibitor. And then later that day, he developed swelling of the face, swelling of the lips. He had the upper extremities got swollen. Just all of these things happened in this patient. He just swelled up. And this is how intense the swelling is. It can actually be life-threatening because you can actually stop breathing. So here's like a patient that probably he has C1 esterase deficiency, probably had no idea, went his entire life like totally fine, or just not even noticing the symptoms, right, of having a C1 esterase inhibitor. And then all of a sudden, doc started him on an ACE inhibitor, and this is what happened. So his face blew up, his lips blew up. Yeah. But this is your classic hereditary angioedema vignette. So that's hereditary angioedema. The other ones, and this is where I said you should know, like DAF and Merle, CD55, CD59, because this pathology state, this is actually a UWorld picture. And I put this in here from my UWorld questions last year. They literally asked, what's the deficient protein? And they listed a bunch of CDs. So it is important to know these. But CD55, this is DAF, and then CD59 is going to be Merle, which I don't even know is on here. Is it on? Oh yeah, right here is Merle. And these are the guys that block complement. So Merle blocks like MAC complex. DAF's going to block C3 convertase. It just blocks parts of the complement pathway from binding to these red cells. So there's a pathology known as paroxysmal nocturnal hemoglobinuria, or PNH. You'll come back to this next year because, again, it has to do with red blood cells, so that's technically hematology. But it's relevant here because of complement. So paroxysmal means it happens suddenly. Nocturnal, it's happening at night. And then hemoglobinuria is basically, this is just going to be like a word meaning that there's hemoglobin in the urine. So this is usually going to, just when you see this word, you know that your red cells are lysing open because hemoglobin is within red cells, right? So a lot of the times when you see anyone has hemoglobinuria, you know that the red cells are being like lysed open. It's not just for this disease. There's other disease states, obviously, where red cells can get lysed open, but this is one of them involving complement. And the reason it happens nocturnally, like why is it happening only at night? Like it's kind of weird. So when you sleep, you actually go into like a slight respiratory acidosis. And this slight respiratory acidosis, for some reason, activates complement. That's the best explanation I could find online of why this only happens at night. Respiratory acidosis activates complement somehow. Don't know how. Doesn't follow one of the 3 pathways, but it's fine. So it's activating complement. And then if you don't have these complement inhibitors on your red cell surface, you're going to get MAC binding. You're going to get lysis of your red cells. So this is a normal person. They have CD55, CD59. It's blocking complement from literally depositing on the red cell and activating lysis. If we don't have these guys, CD55 and CD59, if we don't have these, then complement can bind to the red cell and lyse it. There's nothing else stopping it from doing that. And the other thing I will mention, it's because it's kind of like a biochem tie-in, so I do like to kind of mention anytime there's a biochem tie-in. These CDs, like CD55 and CD59, that's like the purple part and the green part. That's what the CD55 and 59 are. They're linked to the surface of a red blood cell with what's known as a GPI anchor. So sometimes they'll say like, instead of saying like, what is the pathology behind this, or what is the mechanism behind this pathology? So they'll describe PNH, say what's the mechanism behind it. And instead of saying like, oh, there's a deficiency of CD59, CD55, they'll say there's a deficiency of GPI anchored, proteins to like the surface of red cells. So it's just like another way to be aware. They are GPI linked proteins to the surface of the red cell, but that's like another way they can describe what DAF and MERL are. Okay, so, oh, I forgot I did this. Okay, so basically it is a gene defect. So there's a gene, it's called the PIGA gene. That's what codes for CD55 and CD59. So this is genetic. And this is when we just talked about MAC and other complement proteins attack your red cells and lyse them open. And here's how they're going to describe it in a vignette. So they're not going to say patient has hemoglobinuria. They're going to say patient wakes up and sees dark red or brown urine only when they wake up. And then it's fine the rest of the day. So that's like your buzzword for hemoglobinuria, like nocturnal hemoglobinuria. Okay, so then we have to talk about complement deficiencies. So patients can have just straight up complement deficiencies, and it's important to understand like early versus late. They present differently. So an early complement deficiency means it's an early complement protein that's deficient. So usually they say C1 through C4 is deficient. So an early complement deficiency means it's like the early proteins in the complement pathway are deficient. It doesn't mean like it's early in life. It's like the early part of the cascade is deficient. And what they say is early is C1 to C4. It is genetic. So it is a genetic disease. So C1 through C4 are deficient. And how this is going to present, and I've seen both of these. That's why I highlighted both of these like things that you can see. I've seen them both ways. Like they'll describe, they'll ask what the pathology is for both of these. So you have an increased risk of recurrent sinus infections and respiratory tract infections. Hopefully that makes sense because complement is like a part of the innate immune system. It's supposed to kind of get rid of these infections that you probably get every single day. It's trying to get rid of those before you even get sick. So you're just going to be increased risk of an infection if you don't have the complement system. Because if you have a deficiency, that's the other thing, right, about zymogen pathways. It's like if one of these is deficient, the whole pathway is not working because it's like it depends on cleavage of the step before it. And then you also have an increased risk to develop lupus. I have no idea why, but it might come up. I have gotten a question on this before. So your early complement deficiency is recurrent infections, which is kind of broad, but then more specific to it is you have this increased risk of developing lupus. A terminal complement deficiency, so like a late complement protein deficiency, this is anything from C5 to C9. So that's what's meant by terminal. It's like later in the complement cascade. This is also genetic. And you're going to see increased susceptibility of recurrent Neisseria infections. So remember, I did say we needed C3b to kind of to opsonize your encapsulated organisms, like which are strep pneumo, Neisseria, Haemophilus influenza. But you can also see just recurrent Neisseria infections if you have a terminal complement deficiency. And I guess this just tells you more about Neisseria more than anything else. Clearly, you need MAC. You need the MAC complex to get rid of Neisseria infections. And then you can also just have a straight C3 deficiency, like you just don't have C3. So if you, and this is a genetic disease as well. Again, all of these are produced by the liver, so they're all like something had to code for them to the liver to produce it. So just there's a gene, there's obviously a gene defect here. It's not like anything else. It's just another genetic disease. But if you don't have C3, not a big deal. But the issue is you don't have C3b. And if you don't have C3b, yes, you don't have the rest of the complement cascade, right? But you don't have clearance of your encapsulated organisms. So strep pneumo, Neisseria meningitidis, and Haemophilus influenza type B. And these are going to be the guys, these guys are going to be the ones that they'll ask about too, in terms of like complement. And then before we do some questions, this is my notes on complement. Basically, I tried to color code it. It was kind of a mess, but I tried to add every single thing we talked about into one slide. That was kind of the purpose behind doing this. So I really tried to put every single thing on here, even in terms of like the stabilizers, like the anaphylotoxins, like everything. Okay, so this can be kind of like if you're trying to draw out the cascade or whatever, you can kind of like reference it here. Okay, so it is 7. Do you guys want a break or do you guys want to go through questions? What do you guys want to do? How many questions? 1, 2, 3, 4, 5.
Can we get him over with?
Yeah, I'm fine. No break. Let's do it. Okay, you guys want to get him over with? Yeah. You guys will be begging me for a break tomorrow and Friday, so I'm okay with no break today. Okay, so I'm going to give you guys 90 seconds to do these, just like yesterday, even though I do know, like, complement takes a while, like, especially with the test, it takes a while to get, but let's just try 90. All right. Okay, I think most of you guys, it's about a minute and a half. I think most of you guys have answered. So this is a classic example of why I like doing questions, because so many, you guys are split 50-50 on this one. And it's good to kind of see how they're going to present something. So this one is actually E. So this is C5 deficiency. This is a late complement deficiency. And the reason you're like, well, how do I know if it's like C5 or C3, right? Half of you guys pick C3. And you're going to note that because it says the girl has had 5 similar presentations in the past. She's had Neisseria meningitidis. That's the only thing she had 5 times. She's only 6 years old. And she's had 5 Neisseria meningitidis infections. That's more of like a terminal complement deficiency. If I go back to that slide, that's going to be your recurrent Neisseria. Whereas, like, if you have a C3 deficiency, you might have, strep pneumonia one year, you might have Haemophilus infection the next year, you might have Neisseria the next year, you might have multiple Neisseria infections, but not 5 in a row, right? That's going to be more of like a terminal complement deficiency. But again, that's why I think doing questions is so valuable, because how would you have known that, right? If you didn't do a question to see how, like, what they mean by that. So I would say if you see like, and this is like abnormal, yes, no problem. That's why I put it first too. Wanted to get it like out of the way. But this is why too, like, again, 5 presentations of the exact same bug, that's, and when it's Neisseria, that's going to be your late complement deficiency. One of my friends who took STEP like last week said that they asked this like exact question on there. I'm like, oh, perfect. I'll make sure to emphasize it. Yeah, so that's like your late comp. They could have also had like C6, C7, C8, C9. Any of those would have been correct. And here's the explanation. And then, yeah. And then I wanted to talk about here too. Yeah, so, and that's kind of what he's saying down here. Like, yes, if you have early complement deficiencies, you might have recurrent infections, but like nothing limited to Neisseria, like not having Neisseria 5 times. Like if you have a C3 deficiency, of course, you might have like Neisseria maybe once, but more so it's going to be like pneumonia, like the strep pneumonia. It's going to be more something like that. Okay, next one. Okay. Guys, ready to go over this? Any last minute people? All right. So I guess this might have been a little on me, because the patient was started on a new medication. This is not an ACE inhibitor. I should have probably put this as a blood, like it's a blood thinner, basically. ACE inhibitors end in like pril. So if you see anything with ending in pril, that's going to be an ACE inhibitor. But for first year, like you're not expected to know ACE inhibitors. They would tell you something like they were started on a new medication, specifically for the high blood pressure. That's more of how it would present. But you guys did recognize C1 inhibitor protein, which is at least recognized it. But yeah, this would be more so for your hereditary angioedema, which is not kind of how it's presenting. Hereditary angioedema, that's more of like the facial swelling, the upper extremity swelling. This one is PNH. So this is going to be PNH, decay accelerating factor. This is your CD55. So he has a deficiency in this, and that's what's causing these like urinary symptoms and such. His hemoglobin is low, but this is in the blood, and you can think of this because it's being bursted and leaving through the urine. That's why it's low in the blood, because it's leaving through the urine. But this is just like another example of like seeing like they can ask DAF or they could ask CD55. Like they would kind of expect you to know both. Make sense? I'll remember next time to be like, this is not an ACE inhibitor. But even so, they would tell you, they wouldn't say it's for a DVT. They would say like for the hypertension, like they were started on this medication for the hypertension. Okay. So mentioned it here. Okay. Next one. Okay, that was a minute and a half. You guys want to get in your last? All right. Okay, so we got to talk about this one. Guys are a little all over the place, which is good because we're talking about it now. Okay, so what does this patient have? What's the disease? Yes, good. Hereditary angioedema. Good, yes, good. So this patient has hereditary angioedema, so clearly they have a C1 inhibitor deficiency. So that got some of you guys that said serum C1. C1's fine. It's the inhibitor that's deficient. So they're talking about lab testing. They're talking about the CH50 test, basically. So lab testing shows decreased C1 inhibitor levels, so a decrease of which of the following is seen in this girl. So they're talking about CH50. So CH50, we know it's either C3 or C4. So how do you choose between C3 and C4? With this, with a C1 inhibitor deficiency, C1 is going to be like normally active as it is. And what does C1 start the process with? There's two zymogens that Z1 can cleave. C1 can cleave. Yeah, C2 and C4, exactly. So the better test for CH50 would be testing for C4 levels. Technically, C3 would technically it would be low, but let's say we have alternative pathway, right? Like let's say the alternative pathway happens to be active at the same time. It wouldn't be the best test. You wouldn't look for C3 levels for a C1 inhibitor deficiency. You would choose C4. And that's kind of what he says here. Normal role of C1 is to cleave C4, which does eventually lead to C3 convertase, right? But you now consume C4 in the absence of C1 inhibitor. So again, it's that whole idea of like consuming something in the process of in like zymogen pathways. Good, does that make sense? So remember C4 is better here. If they had C2 and C4, you wouldn't be able to pick, well, you might be able to because CH50 doesn't look at C2, but I still don't think they would do that. It's usually going to be C4 with this C1 esterase inhibitor deficiency. Okay, two more. Okay, you guys ready for this one? Seems like a lot of you have answered. Okay. So this guy, strep pneumo, you should be like, that's one of the encapsulated organisms, right? And he's only 3 years old. He's had several bouts with this just over and over again. And they're like, oh, there's a complement that's depressed. So this one is going to be C3. So again, no C3, no C3b. You can't opsonize encapsulated bacteria. You can't get rid of it. This is an encapsulated bacteria. So that's that. C5, remember this is your Neisseria. This is like your recurrent, but it could be C6, C7, C8, C9. Yeah, any of those could also be with your Neisseria. And then everything else is just kind of an early complement deficiency, but C3 is specifically for your strep, your Haemophilus, your Neisseria. Okay. And then this last question is a little tricky, but I want to see how you guys do with it. It kind of ties into some stuff we did yesterday, but I want to see how you guys do with it. I think when I wait too long, you guys start changing your answers, and I'm like, no, don't change it. Oh, okay, go ahead. Go ahead. Okay. Okay, good. All right, so we're gonna talk about this one. So this patient has lupus. We're gonna talk about lupus. I think like the last day. This is how lupus presents. But even if you didn't know that, you could read this and be like, okay, she has a fever. Like she's got inflammation happening, like just tons and tons of inflammation. It's warmth, erythema, swelling. This is like all the cardinal signs of inflammation, right? We got tons of inflammation happening. So a lot of you guys did say that IL-1 was increased, good, and TNF alpha was increased, and that's good. And I know a lot of you guys were like, "Oh, I'm not sure about TNF alpha." TNF alpha is going to be increased with inflammation. It's one of those ones. It's one of the acute then recruit cytokines that's released during the basically the innate response to activate inflammation. So anything where IL-1 and TNF are not increased get thrown out basically. And if you guys didn't know that, that's fine. Like in terms of didn't know about TNF alpha, that's fine. You could have done this with IL-1. And you know that IL-1 is definitely going to be increased regardless because she has a fever. And remember, IL-1 is hot, you know, the hot and hot T-bone steak. Okay, so we've narrowed it down to basically, what, 2? Yeah. Yep. And a decent majority of you guys were between these two, which is good. Okay, so next is talking about C3. So this is the part that was kind of tricky about this, and it's kind of like, sorry. But lupus, one of the things that happens in lupus, and I think I do have it on one of the slides, that's why I threw this in here, but you get antigen antibody complexes. So you get these complexes of antigen and antibody. They kind of circulate around the body, and that's what kind of causes these pathology states. Anytime you get antigen antibody complexes, you get activation of complement. And that's just always going to happen regardless. So we're getting activation of the complement system. We're getting binding of like C3b to these things to help clear them out of the system. So since complement gets activated, it means it's going to get used up, meaning the correct answer here is C. So we're getting a decrease in C3 levels because we're going through the complement system. So we're using up complement component C3. I know a lot of you guys, and some of you guys switched it from C to B, and I'm like, no. But I think that's just what happens when like you're looking at a question. But does that make sense? I know it was tricky. I did put this on here. It is tricky. So if you're like, I did not know that there's antigen antibody complexes in lupus. Maybe you didn't know that yet, and that's fine. But all I'm trying to show you is like how they kind of throw in complement with like inflammation. And usually when there's inflammation, I have to be careful how I say this, but it's like usually with inflammation, but a lot of the time it's with these antigen antibody complexes, which is a lot of the time what causes inflammation. You're gonna use up complement. I just really want to emphasize this as we keep going through. If complement is decreased, we're using complement. Yeah, the CH50 test, it'll get you. So I just really want you guys to keep seeing that and just keep reminding yourself of it. If I have lower levels of C3, I'm going through the complement pathway. Same with C4, same with C2, same with any of those. Yeah, you guys won't get it wrong on the exam. Yeah, you definitely won't get it wrong on the exam. Okay, but does that make sense? I mean, as well as you would know lupus, but inflammation, decrease C3. It's going to basically cause up. It's just going to cause he's saying it's famous as a disorder causing low serum complement. It's because all those antigen antibody complexes just bind up all the C3b and it just uses up all of that complement. Okay. Let me stop sharing. Stop recording.